This video is going to focus on step 5, which is all about virulence factors. And so virulence factors are going to be the strategies that the pathogen employs to weaken the host, right? Because they're trying to take over so they can replicate and then eventually exit the body and find a new host. And so they are going to have to kind of attack our body, our cells and things to try and kind of take over. And so we're going to talk about bacterial virulence factors, and then we'll talk about some viral virulence factors as well. So when it comes to bacterial virulence factors, again, these are going to be approaches, strategies that the pathogen is going to use to cause disease at the cellular level. Okay, so these are not signs and symptoms. It's not, oh, they're going to cause a cough, right? No, we're saying what's happening at the cellular level. And so we're going to talk about 5 common virulence factors. And so on your main PowerPoint that's already posted, there are only 4, but I went ahead and added a 5th, even though we're going to talk about it again at a future step. But let's go ahead and start talking about these various common forms of bacterial pathogenesis. So again, strategies they use to be pathogenic and to cause disease. First up are exotoxins. And exotoxins are protein molecules. Typically, they are enzymes, and they are very, very potent. Okay, they normally have a very low LD50, meaning that very few of them are required to actually be lethal to 50% of the population. And because exotoxins are enzymes, they have a very diverse mechanism of actions. Okay, so the exotoxin produced by Clostridium tetani, which causes tetanus, is a neurotoxin. But what it does is it causes muscle, like rigid paralysis. It causes muscle contractions. And so essentially people will eventually inhale, but then they can't relax. And so they cannot exhale. Okay, but a different Clostridium species, Clostridium botulinum, which causes botulism, is also a neurotoxin, but it actually blocks the release of acetylcholine, so it causes flaccid paralysis. So it's essentially the opposite. Your muscles relax, and so you'll exhale, and then you can't recontract to inhale. Okay, so both result in death normally, pretty horrible ways. But those just 2 examples of different protein exotoxins that target similar pathways but have different mechanisms. There are also toxins that target translation. There are different exotoxins that damage blood capillaries under the skin, causing a rash. Others target the ciliary escalator, and so it causes mucus buildup, and that kind of leads to whooping cough. And so just exotoxins are very, very diverse. What they have in common is that they are very potent, they are proteins, and they are actually proteins that are made within the bacterial cell, but then they get secreted from the cell. Essentially that bacteria makes them and then ships them out into the host, where it goes and wreaks havoc on the host cell. Okay, so they're very, very dangerous. So another thing I want to mention with exotoxins is because they're proteins, they're very good triggers of the immune system. And so our body will actually make antibodies against these exotoxins once we're exposed. And so we call these specific antibodies antitoxins because they're targeting a toxin. And so it turns out, like in the lab, certain exotoxins can actually be modified in order to produce toxoids. And toxoids are weakened toxins that are not going to wreak havoc on the host. And so these can be used as vaccines. So toxoid vaccines, they've lost their disease-causing properties, okay? And so, but what it's doing is it will, for instance, introduce our body to an inactive form of the tetanus toxin. So the overall structure is normally maintained, but again, it's inactive. It's not going to give us tetanus. But that way, if we ever step on a rusty nail and we do introduce Clostridium tetani into our body and it releases the toxin, we already have antibodies that are going to recognize that 3D shape of the toxin so it can be eliminated before actually, causing chaos within the cells. So the antibodies specific for exotoxins are called antitoxins. And so as kids, we get the DTaP vaccine, and the D is for diphtheria, which has a potent exotoxin, and the T is for tetanus. And that's just two examples. Okay, so we're going to contrast that with endotoxins. And so a very common endotoxin is lipopolysaccharide, which is present in the cell wall of all gram-negative organisms. And so endo means within. So an endotoxin is made within the cell and it remains as part of the cell. Okay, so unlike the exotoxins, which got shipped out into the host. So with the endotoxins, they all have a very similar effect. Okay, they don't have a diverse mechanism of action. Okay, they all have a very diverse, they have the same symptoms. They cause the same symptoms in a host because LPS is LPS. It doesn't matter what gram-negative organism has it, it's still the same molecule. So it's going to cause the same symptoms in anybody that gets infected with a gram-negative organism. Okay, and again, that's very different than exotoxins that had the diverse mechanism of action. And so we say exotoxins are specific. The effects of the exotoxin are specific to that particular pathogen, right? Tetanus caused rigid paralysis, whereas botulism specifically causes flaccid paralysis. So the mechanism of exotoxins are specific to the disease. The endotoxin mechanism is the same for all different organisms. And they typically cause like fever, malaise, which is like aches and pains, and fatigue, but it can lead to shock and death, if it gets, like, people get really, really sick from a gram-negative. There are no endotoxin vaccines available. And actually with antibiotics, if you take an antibiotic to treat a gram-negative infection, sometimes the doctor or nurse will tell you, you're actually going to feel a little bit worse before you get better. And that's because when the antibiotic kills the bacteria, it's releasing all of those LPS into like the bloodstream in the body. And so you're going to really feel the effects of those endotoxins once the antibiotics start to work. But eventually they do get cleared from the body and we start to feel better. All right, the virulence factor that I added is the capsule. Remember, that's the sugary glycocalyx. And it is used to evade phagocytosis. And so it is mentioned, I believe it's step 6, the evasion strategies, but I really wanted to just plant the seed now that the capsule is a virulence factor. Because it's able to evade phagocytosis, that is a weapon that it's using to kind of prolong the effects of these other virulence factors like an exotoxin or an endotoxin. Another virulence factor employed by bacteria are going to be exoenzymes. And so these are enzymes, again, made by the bacteria that are then shipped out into the host cell. And again, very diverse MOAs. And I do want you to know the exoenzymes that are present. I believe it's on slide like 24 of your PowerPoint. So we're also going to talk a little bit more about these again with evasion, but some important exoenzymes cause clotting, And that's going to allow bacteria to hide from our immune system. Some of them are going to lyse red blood cells, which are actually going to promote the spread of the pathogen. Others are going to, again, break down that hyaluronic acid. So we've already talked about hyaluronidase. And then catalase is another one that we've talked about. That helps break down toxic peroxide. So again, these are just strategies used by the bacterial pathogen. Okay, and then the last one that I wanted to mention is that the pathogen will sometimes stimulate the body's natural defenses, but it does so in such a way that it causes damage, which is going to disrupt how the body typically functions. And you don't have to memorize all the examples that are in the PowerPoint, but I wanted you just to kind of see what it means, how it kind of turns the body against itself. So for instance, with pneumonia, right, that's going to be targeting the respiratory tract. And so you tend to get bacterial infections in the lungs. And so the presence of a pathogen in the lung is going to trigger some of our immune cells, our phagocytes, to come to the lungs and try and clear that infection. But those bacteria that have the capsule, right, they're able to evade this process. And so because the original set of phagocytes are having a hard time eliminating those pathogens, more phagocytes arrive to try to help them out. And so what then happens is the inflammatory response is triggered, right? Because we need to keep these pathogens in this particular area. And so as the inflammatory response is triggered, it actually leads to this accumulation of fluid in the alveoli. And when they become filled with fluid, the normal functioning of gas exchange is going to be impaired. Okay, so it's kind of making our body not function as it should. And you see this also with meningitis, which is swelling of the meninges, which are those membranes that surround the brain. And so when the meninges become inflamed, right, you're experiencing increased pressure in the cerebrospinal fluid and the brain. And so it can result in things like neck stiffness, sensitivity to light, drowsiness, fever, and even things like confusion. Okay, so again, just the normal parts of our body are not acting as they normally would. So on the next slide is a table where we're going to compare exotoxins and endotoxins. Okay, so we're going to go through this kind of quickly because I just talked about most of it. Exotoxins are significantly more potent than our endotoxins. So when we're comparing the lethal dose, the LD50, exotoxins have a much lower LD50, meaning that it takes less of them to kill 50% of the hosts. So that means low LD50 means they're much more potent. So therefore, endotoxins are the opposite. They are high. The effects on the body varies, okay? Because again, they're so diverse. Each exotoxin acts differently. Another thing to say here is that the effects are specific to the disease because each pathogen has its own specific, well, if the pathogen makes an exotoxin, that exotoxin will be unique to that pathogen and therefore unique to that disease. Okay, so it has a specific effect depending on the pathogen. Endotoxins have the same effects on the body: fever, malaise, fatigue. It can eventually be things like diarrhea, shock, or death, but those are the shock and death are in extreme cases. With our chemical composition, our exotoxins are protein, but because the endotoxin is LPS, the L stands for lipo, the PS is polysaccharide. So this is a lipid carbohydrate combination. Okay, so it's not going to be susceptible to certain environments like proteins are, because remember, proteins are in fact subject to denaturation if things get too hot. Whereas the lipopolysaccharide is not going to denature if the temperature increases inside the body. Okay, so remember, think of like a fever. Okay, so we're not talking super extreme heats here, okay? So we did mention that exotoxins are able to form toxoids, whereas LPS cannot, because lipids and sugars are just not strong triggers of the immune system, and so therefore we don't create those antibodies against them. Okay, so then this next one was just kind of redundant because toxoid formation has to do with the immune response. So when it comes to fever stimulation, would it make sense for a protein to stimulate a fever? No, it wouldn't, because the fever is going to be increasing the temperature, right, which could lead to denaturing the exotoxin. The pathogen does not want its own exotoxin to be denatured, right? That's the weapon that the pathogen is using. So the pathogen that makes an exotoxin does not want to stimulate a fever, because that runs the risk of destroying its own weapon. But endotoxins, because they aren't heat sensitive, typically do stimulate a fever. When it comes to manner of release with exotoxins, they are made within the cell and then secreted outside. A lot of exotoxins are what we call AB exotoxins, and that means there are two components of an exotoxin, the A component and the B component. The B component is the binding component. That's the part of the exotoxin that is going to bind to the host cell and going to trick the host cell into taking in that toxin. Once the AB exotoxin gets inside of the host cell, the two components separate. B gets recycled and sent back to the bacteria so it can be used again, and the A component is the actual enzyme that then goes and wreaks havoc on the cell. The endotoxin, I put none, but I kind of want to rethink that. So what I was trying to get at by saying the word none was that the endotoxin remains a part of the intact bacterial cell. But the endotoxin is released throughout the body when those gram-negative cells are killed. So you might want to put next to the word none, put lysis, because once those bacterial cells die, maybe they become leaky, maybe they are lysed. Then the endotoxin is spread throughout the body, and that's when we get the fever, the malaise, the fatigue. So go ahead and make that little note next to the word none. And then our typical sources, exotoxins are predominantly going to be made by gram-positive. Okay, so mainly gram-positive, but there are a few gram-negatives that do make exotoxins, but it's not very common. And that's because gram-negatives already naturally come equipped with LPS. They've already got an endotoxin, so they don't normally need an additional toxin. Okay, so exotoxins are typically gram-positive, whereas endotoxins are always gram-negative. All right, oops, sorry, my animation here got wonky, so let me just kind of put all of those up there. I apologize for that. Okay, so for viral mechanisms of pathogenicity, one approach is the release from the cell, because with lysis, right, it completely destroys the host cell, right? So that is a strategy of weakening the host. It's killing our individual cells. So we call that a cytocidal effect. So cytal meaning killing. So it will kill the host cells. But our enveloped viruses have two different options for release. Either it's going to bud away from the host cell, or it's going to be released via exocytosis. But both of these, if they happen too much, they're constantly pushing and weakening the plasma membrane, which is dangerous for the host cell. So these two exit strategies tend to have a more cytopathic effect, which can still lead to cell death, but it definitely harms them because it's disrupting the integrity of that plasma membrane. A second virulence factor or strategy that some viruses use are going to be if they employ the lysogenic or latent cycle, because by essentially just hanging around in the host, right, the viral DNA gets incorporated into the host DNA. And so during transcription and translation and other normal host cell processes, some of the viral DNA is getting transcribed and translated. And so this can result in functional changes to the host cells. For instance, some viruses cause normal hormone production to cease, right? So that's not any good. And in other examples like HPV, human papillomavirus, which causes genital warts, it can lead to cervical cancer because it messes with mitosis. It causes it to go unchecked or unregulated. And so that can lead to oncogenic or cancerous circumstances. And then finally, syncytium formation. And so with you can see syncytium occur, which is cell fusion. You see this with some enveloped viruses because some enveloped viruses are able to fuse with the host membrane as a way to get inside the host cell. These enveloped viruses have what's called a fusion protein. It allows the virus to fuse with the host cell. And so as the virus is being replicated, this fusion protein is made and expressed at the host's plasma membrane, but it can actually cause neighboring host cells to fuse together. And when that happens, it causes individual cells to fuse together, forming one large multinucleated cell. And that's what's referred to as the syncytia. And when all these cells have fused together, right, they kind of lose their individual abilities and functions. And so it's disrupting the normal function of the individual cells and even the tissues, right? Because tissues are made up of certain types of cells. And if all those cells start fusing together, then the tissue isn't going to function as it should. All right, so that is going to be the end of this video. We've got, I think, 2 more videos left for this section. Let me know if you have any questions.